All right, so you have already learned that water has some very special properties, and that's what makes it so important, especially to living things. Now we're going to take a look at the movement of water across the cell membrane. So, because water is so important and so special, the movement of water across the cell membrane gets its own special term, and that's osmosis. So osmosis is the diffusion of water from a higher concentration of water to a lower concentration of water across a selectively permeable membrane. So we use a lot of things that kind of look like this, and the blue part represents water. Any little colored dots would probably represent solutes, and this little dotted line represents a selectively permeable membrane. That means some things can get past, but some cannot. So if we look at this a little bit more closely, this side doesn't have a whole lot of these little green dots, so there's not a whole lot of solute concentration. Water is the solvent. Sugar is the solute in this example to make the solution. On this side, water is the solvent. The little green dots represent sugar, which is the solute, to make a higher concentration of sugar water on solution on this side. So we're at this little selectively permeable membrane. Remember, there's less concentration of sugar on the left, more concentration of sugar on the right. So the little green dots, the little green blobs represent your solute, your sugar, and of course these are all your little water molecules. On the left-hand side, there are not as many solute molecules as there are on the right-hand side. The solute molecules have a sort of partial attraction from the polarities between the water molecules and the solute of the sugar. So these little water molecules are attracted to this sugar, to this solute. Same thing on this side. These little water molecules are attracted to this solute, attracted to this solute, attracted to this solute. Notice that because there's more solute molecules on the right-hand side, that there is not a whole lot of free water that's left. These solute molecules are kind of taking up the water molecules' attention. So, since the water molecules are paying all of their attention to the solute, there aren't as many free water molecules to move back and forth across the membrane. There are, however, some free water molecules on this side, and these guys are going to move back and forth across. There's a lot more free water molecules on this side, however. So as they move back and forth across, chances are, because there's more solute on this side, they're going to end up staying over here because they're going to pay attention to these little sugar molecules. My selectively permeable membrane is allowing this water to move across, but the sugar molecules, the solute molecules, they're too big, so they can't get through the gaps in that membrane. Overall then, osmosis is going to occur from the left side to the right side. So the overall movement of water, the net movement of water, will be from the right side where there's more water concentration to the left side where there's less water concentration. There's more free water on the left, less free water on the right, so eventually more water is going to move to the right-hand side. The direction of osmosis is determined by comparing. You need to compare two solutions. So when we use words like hypertonic, hypotonic, or isotonic, we have to say that a hypertonic solution has more solute than another solution. A hypotonic solution has less solute than another solution. So we're always comparing. Isotonic means the two solutions both contain the same net amount of solvent or net amount of solute. Because this solution is hypotonic and this solution is hypertonic, Osmosis is once again going to occur from the left to the right. There's more free water on this side. Less solute means it's not attracting as many water molecules and keeping their attention so they're able to move. There's more solute on this side, so as those free water molecules move across, they end up being attracted by that sugar solute, and they end up staying over there. So these solutions can actually affect cells in real life, and they can have consequences. If I put a red blood cell into a hypotonic solution, the erythrocyte is hypertonic to the solution. The solution is hypotonic to the red blood cell. So I put a red blood cell into pure water. The pure water solution is obviously not going to have any solute in it, and so it's going to be hypotonic. So because the hypotonic has more free water, and the hypertonic solution in the red blood cell is going to attract that water, water is eventually going to move into that red blood cell, and it's going to swell, and eventually it's going to lyse or burst. Same thing for a plant cell. I put a plant cell into pure water. The solution that I put the cell in is hypotonic. The cell is hypertonic, and water is still going to move into the cell. Plant cells actually like that because while the animal cell will lyse or burst, the plant cell has a cell wall. And so the cell wall kind of pushes back as the water is expanding the cell and pushing outward. And this is a benefit to a plant cell because it gives it a turgid or normal situation for the cell. And so these plant cells help the plant to stand up straight because it's got that pressure against those cells causing it to stand up straight. If I put that plant cell into an isotonic solution, that means this solution has the same amount of solute as the cell. The water is going to move in and out of the cell in an overall net equilibrium movement, net equal movement of that water. And so instead of building the water up on the inside and pushing against that cell wall, the vacuole is going to shrink because it's going to lose some water, and this actually is going to make the plant wilt because the cells don't have that pressure against them to hold them up. Animal cells, however, love isotonic solutions. This is ideal for animal cells. In a hypotonic solution, the pure water is going to move into the hypertonic cell, and so it's going to swell and burst. But in an isotonic solution, the solute of the solution is the same as the solute concentration of the cell. So water is going to move in and out and continue to move in and out, but at an equal rate. So there's a net movement of zero of the water, even though it is actually moving. And it's a dynamic equilibrium. It doesn't ever stop moving. Neither cell likes to be in a hypertonic solution. So you put a red blood cell into a solution that has too much solute, and the water is going to be pulled out of that cell, and it's going to cause that cell to shrivel up. Same thing for a plant cell. You put a plant cell into a hypertonic solution. Because the cell itself is hypotonic, the water is going to move out of the cell, and it's going to show plasmolysis. So here's some actual microscopic cells showing a red blood cell in an isotonic solution. This red blood cell is in a hypotonic solution, so there's more water in the solution and there's more solute in the cell. So it's swelling up. This is a red blood cell in a hypertonic solution. The solution that it's in has a lot of solute. The cell itself is hypotonic to the solution, so the water is moving out of the cell and causing it to shrivel up. Plant cells like to be in a hypotonic environment. The cell wall, remember, it provides a structural support. That helps to even out, as the cell swells and pushes against the cell wall pushes back, and so they're adapted to exist in an environment that includes a hypotonic solution. They do best because the turgid pressure keeps those plants upright. Here's an example of a cell that is being placed in a hypertonic solution, so the water is being pulled out, and the cell is going through plasmolysis. Another actual microscopic picture, little algae cells that are going through plasmolysis. This is how they should look, and this is how they look when they're placed in a hypertonic solution. All right, animal cells. So this is an example of a protist in freshwater environment. So it might be like in a pond or something like that. The environment that it's sitting in, this freshwater environment it's sitting in, is hypotonic to the single-celled creature. Because of that, this water in its environment is constantly entering its cell. Well, that would cause this thing to swell and burst, which would cause the little paramecium, would cause the little paramecium to die because it lyses. So it has a special little structure called a contractile vacuole, and this gathers up that water, and then it releases that water back into the environment. But because that water is going from an area of lower concentration to an area of higher concentration of water, this contractile vacuole moves the water against its concentration gradient. So it needs energy in order to do that. It requires energy in order to work because it's moving the water against its concentration gradient. Little tuna. This little tuna is actually in a very hypertonic environment. It's in salt water. And so the water is constantly trying to move out of the fish into the saltwater environment. The tuna needs to adapt, and so it has to take in lots of water continuously, and it has to pump out the salt. So it's adapted because it's got special cells in its kidneys and special cells in its gills that's able to concentrate that salt and release that salt back into the environment. All right, so isotonic solutions. Isotonic solutions mean that we're eventually going to come to an equilibrium where we have as many solute particles on one side of the membrane as we have on the other side of the membrane. But as I mentioned before, this is a dynamic equilibrium. These molecules, they're still moving back and forth across, but the overall net movement would end up being, it's zero. So it's not moving more one way, more moving the other way. They're moving equally back and forth across. So it's a dynamic equilibrium, but these molecules don't stop moving. When we talked about diffusion, we mentioned the fact that diffusion occurs along concentration gradients, and it occurs along each specific solute's concentration gradient. So in this very first picture here, we have 2, 5, 6, 7, 8, 9, 10, 11, 12. We've got 12 yellow solute particles. We have 6 purple solute particles. So obviously there's more solute on the left side than there is on the right. However, each one of these types of solutes, they're going to move independent of each other. They move along their own concentration gradient. So because there's more yellow on this side, it's eventually going to diffuse so that the yellow is equally distributed, but its net diffusion is going to be from the left to the right. The purple molecules are more concentrated on the right. They are going to diffuse independent of the yellow solute molecules. So they're eventually going to diffuse so that they're equally spread, but their net diffusion is going to be from the right to the left. Solutes are going to diffuse according to their own concentration gradient. They do that independently of any other solute that's present.